Hello and welcome to the webinar on the identification and organic management of cucurbit viruses by John Murphy of Auburn University. This webinar is part of a series of 3 webinars organized by members of the NIFA OREI-funded Eastern Sustainable Cucurbit Project, which is a collaboration of growers, researchers, and extension agents working to find solutions for the many challenges facing organic cucurbit producers. This is your host, Alice Formiga of the eOrganic Community of Practice at extension.org. Today's presentation will last about 45 minutes, followed by 30 minutes for questions. Today I'd like to welcome Dr. John Murphy. He's from Auburn University in Alabama. His work focuses on plant virus ecology, how plants spread and develop resistance to viruses, and developing organic management strategies for insect-borne viruses. So with that, I'm going to hand over the screen controls and the microphone to you, John.
Thank you very much, and welcome everybody to this webinar, and thank you very much for participating. So today, my plan is to give a bit of an overview on plant viruses and how they cause infection and spread in the field, a bit of an introductory plant virology session, maybe for half of the talk. Then the other half, I will be focusing on the 4 viruses of our project, along with the management strategies that we have tried in the past. So this project is sent through the USDA and Michael Mosaic at Cornell University as a project leader. So a little bit about plant viruses as we start here. Plant viruses threaten just about every crop. If you have whatever crop you have in the field, there is a virus that has the potential to cause losses to your crop. Now, a lot of what I'll be talking about are field-based issues here. So if you have housework, it might be a little bit different. But there are plant viruses everywhere, and no matter what kind of production system you use, whether it's organic or conventional, you will be running into some plant virus issues. Most of the time, it's the low incidence in the crop. Maybe a plant here, a plant there, you'll see some virus symptoms, but you rarely will have a severe outbreak. If you do, it requires the alignment of 3 major factors. One is you need a large source of virus to build up over time, perhaps in the native plants or weeds around your crop, to be brought into your crop. You need the vectors available to deliver the virus to the crop, and then you need a vulnerable crop that can sustain all this infection. So the vulnerable crop part is something where if your crop is young at the time when the virus is spread into the field, that's usually when you have the greatest amount of disease, and we'll talk a little bit more about this as we go along. Management of viruses is particularly difficult, especially if they have insect vectors that fly around. If you have resistant varieties, it's great. We do not have cidal sprays like fungicides or nematicides for viruses. And we often try to come up with plans, practical plans for growers to reduce virus losses, and they tend to focus on the vector. Okay, shifting here. So I'm going to talk a little bit about viruses themselves and how they cause infection. So viruses are pretty simple organisms. They're actually considered them infectious agents. They come in different sizes and shapes. For instance, these are rigid rod-shaped particles, flexuous rod-shaped particles, icosahedron particles, or spherical particles. One thing that is, while they're all different sizes and shapes, they all are ultra microscopic, which means that you can only view them with an electron microscope. They are simple infectious agents. They consist of a coat of protein subunits that encode or encapsulate the nucleic acid of the virus. And that nucleic acid may be RNA or DNA. Viruses are obligate parasites. This is an important point for our talk today. They require a living cell and host to perform their infection cycle. They use the host material for building their proteins and genetic components, as well as the host machinery, in many cases, for producing more viruses. They do use their own genetic material as a template, but they are totally dependent on the host, so they're obligate parasites. The infection process of a virus is pretty fascinating. If you think about a leaf and you take a cross-section of that leaf, this is the drawing in here. Leaves typically have upper and lower epidermal cells. In the middle are various forms of mesophyll cells, and embedded within that is a vascular bundle, which consists of phloem and xylem. And you need to remember something about plants is that plant cells have a rigid cell wall around each cell. And within that is the plasma membrane that encloses all of the living content of the cell. So when a virus is introduced to a leaf, say by its vector, it has to be introduced through the plant cell wall, through the plasma membrane, and into that cell without killing that cell, because the virus is an obligate parasite. So you have to have a non-lethal wound to introduce that virus into that cell. Once in that initial cell, the virus starts replicating very quickly, producing its proteins, replicating its genome, and starts spreading to all of the neighboring cells. Now, for plants, cells are all connected by way of rather complex channels called plasmodesmata. And every cell is connected to its neighboring cell through at least one plasmodesma or plasmodesmata. Virus gets into each neighboring cell, and its infection cycle just begins all over again. It's a little machine that replicates and produces many, many forms of itself. And this continues to occur cell to cell. Eventually, it gets into the vascular bundle, where in most cases it will end up in the phloem. Now, if we go over to this plant here, and let's just say where this X is, where the infection occurred, the virus spread throughout that leaf. Once it gets into the phloem, it leaves that leaf and spreads sometimes rather rapidly through that plant, most commonly initially to these younger tissues. So with plants, there is a directional flow of the phloem called a source to sink flow, where mature leaves that are photosynthetically active produce photoassimilates that are taken up by the young developing leaves and roots, fruits. And so the virus goes in the direction of that flow of photoassimilates, initially moving to the young parts of the plant where it invades those leaves, and in time also invading the more mature leaves. And that movement to these young leaves is where we get very typical virus symptoms, this mosaic type pattern, this light green, dark green type of disease symptom. So once in the plant, the virus remains intracellular. It never leaves the cells of the plant. It just moves cell to cell and throughout the plant. The result is, if the virus was introduced into a leaf, sometimes you can get lesions at the site of infection. So these are chlorotic and necrotic lesions. If they're called local lesions, that means that the host has been able to localize that infection to a few cells and the virus does not spread. And this is not a problem for us agriculturally. The big problem comes when we have systemic invasion of those young tissues and the virus is able to spread throughout the plant. And depending on the virus and the plant and the time of infection, we get various types of systemic symptoms and leaf deformation. If a plant is infected early, we end up getting stunting of the plant, and interestingly, these stunted plants often have the same number of leaves as an unaffected plant, but the leaves are smaller and deformed. Now, this kind of plant will not produce marketable yield, and it serves as a source of inoculum for other plants early in that infection state. So that's a real problem. But the virus can move into fruit and cause discoloration and misshapen fruit, smaller sizes, numbers, and of course that's a problem if you're trying to market your fruit. So how does a virus move around from plant to plant in the field? Well, I'm going to mention just briefly mechanical and grafting, but I'll spend a little more time on seed transmission and transmission by biological vectors. So mechanical transmission is where if you're pruning plants or staking and tying plants, there's a chance that you can wound the plant and transfer the virus to another healthy plant that way. If plants have like hairy leaves with a lot of trichomes on the leaves and they rub together, sometimes you can get transmission that way. In most cases, the virus has to be a pretty stable virus, so it's in a genus that's a rather stable type of virus for this to happen. but it can happen in nature. Grafting is a way for viruses to move from the plant, sometimes purposely by generating grafted plants or through roots in the soil. Sometimes the virus can be transferred from one plant to the next. Seed transmission is a big problem. So virus can be transmitted through seed. Not all viruses can be transmitted through seed, and if they are transmitted through seed, they are not done so in all plant species or varieties. But if a virus is able to be transmitted through seed, the problem is that it is in that plant from the beginning of development of that plant. So if we think about having this field here and these little X's where there are infected plants that were infected right from the beginning of time because it's seed transmitted, we have a random distribution of virus in the field because of seed transmitted. These plants that are infected early are a more severe disease. They are now a source of virus if vectors come into the field. And if this occurs early in the season, then these other plants are also young. So if the vectors come in and spread this virus, you're going to have a crop that's going to be severely damaged with very yield coming out for you. So transmission is a very, very big problem that is concerning to many people. Biological vectors include insects, mites, fungi, nematodes. Now, if a virus is transmitted by, say, a nematode, it is not transmitted by the other type of vectors. The virus-vector relationship is very, very specific to that virus and that vector. So they're not transferred from plant to plant by some other type of organism. I'm going to focus on insects, and insect vectors are a problem, well, because they fly. So they can transmit virus quickly within a field, among fields, over regions sometimes, depending on the virus and the vector. They also add a complicating dimension to trying to manage viruses because we have virus potentially flying into the field. So some of the insect vectors include whiteflies, which there's a growing number of important viruses being transmitted by whiteflies, leafhoppers, beetles. Many of the legume viruses are transmitted by beetles. Thrips, interesting, they transmit a genus of viruses called tospoviruses. Tomato spotted wilt virus is included in that. This is one of the few virus groups that is able to infect, actually replicate in its insect vector. So it infects the vector, and it also is able to be transmitted to a plant where it infects the plant. In most other cases, that is not the case. The vector just acquires the virus and transmits it, but the virus does not replicate in its vector. Aphids are the vectors that transmit more viruses than any of the other vectors. And this is where we're focusing our studies on our aphid transmitted viruses. So aphids have a piercing, sucking, feeding behavior where they have a syringe-like feeding mouth part, if you'll call it that. And this syringe-like mouth part can create non-lethal wounds in the cell. So when an aphid lands on a leaf, it initially will probe an epidermal cell to see if it's a suitable host to feed on. It might walk along the leaf and probe several times just to determine if it's a suitable host. That probing mechanism is important for some viruses, and the viruses that we work with, in fact. So we'll talk about that in more detail. Once the aphid feels that the host is suitable, it'll settle in and it'll run its stylet between cells down into the phloem, and it'll sit and feed on the phloem to feed on the sugars and materials that are transported in the phloem. So I want to talk just briefly about diagnosis. I know I'm hopping around a little bit, but all of this will come together eventually. So diagnosis of virus disease is somewhat complicated, although initially everything is done based on visual. So you walk in your field and you see a symptomatic plant. It looks like it might be virus, and so you understand the type of symptom, the severity. You might determine the number of plants in the field, their distribution. Are they along a field margin? Are they randomly distributed? That's not so bad, but it's very difficult to try and determine which virus is the causal agent of that disease based on symptoms. So for instance, two unrelated viruses might cause very similar symptoms. In this case, this cucurbit plant, cucumber mosaic virus and watermelon mosaic virus, two different viruses. cause very similar symptoms like this in cucurbits. One virus species might have strains that induce very different symptoms. This is a black-eye cowpea mosaic virus and cowpea, two very closely related strains of the same virus species, and yet it induces very different symptoms and a very different degree of severity in the disease. Mixed infections of two or more viruses occur very commonly in the field. This is within single plants. So I have this plant here as an example. This is potato leaf roll virus in potato. Alone, potato leaf roll virus is pretty easy to diagnose. It causes pretty distinct leaf roll symptoms in potato. This particular plant was also affected with 3 other viruses. So potato leaf roll virus was masking the symptoms of the other viruses. We didn't know that. In other cases, mixed infections can lead to reducing symptoms. They can get this masking effect, or you can have a synergistic interaction where the symptoms are worse than either virus alone. On top of that, one virus may produce different types of symptoms depending on the plant species, the variety, the age of the plant at the time of infection. So basing diagnostics on symptoms is quite complicated. So what process can you use if you have virus? So you see that there are some virus symptomatic plants in the field, and maybe you just need to say, yeah, I have some infected plants. There's just maybe a very small percentage of plants, maybe a random plant here and there, and that's all you really need to know. That's okay. You may want to go to the next level and know, well, what is the genus of that virus? Because at least if I go to the genus level, I'll have some idea about its vector, and I might be able to start planning for management strategies if the incidence of the virus continues to grow over time. You can also take that down even to the species level, which also will give you information on the vector, but you may have purchased seed that is resistant to one virus species in a genus but not others. You might want to know if that source of resistance is holding up in the field. So you can, depending on your needs, you may want to go all the way down to the species level. How do you have this done? Well, there are testing services at agricultural universities. They have diagnostic labs, and there are commercial companies available. You can go plant virus testing labs and find them. There will be costs associated with this, and those costs vary. There are 2 basic approaches that are used today for diagnostics. One is a serology-based test, where an antibody is generated to the virus, and using this approach, there are some rather simple field-based tests that can be used, or you can have something rather complex. If you have one important crop and the sample you want to send in needs to be screened for 25 or 50 different viruses, a company can do that, but it will be expensive. So the prices range from a few dollars per sample up to hundreds of dollars depending on what you need. The limiting factor here is the availability of an antibody. In order for someone to go to the trouble of generating the antibody and developing the kit or the test, there has to be a virus that they think they will make some profit on. So sometimes there are new viruses that show up that we don't have serological kits to. In that case, you might go to a nucleic acid-based test. These tend to be more technical and costly. They're not used for surveys like they would a serology test. But the potential here is that today we have sequence information on the genome of just about every virus there is. You can get that sequence information, look it up, and then generate the test that you need to do to detect just about any virus there is in the world today. So that's a very powerful approach. So I'm going to switch a little bit here to the project that we're working on, and I'm going to tell you a little bit about the viruses that are involved. One virus is cucumber mosaic virus. This is a fascinating virus. It has an icosahedron particle. Actually, it has 3 particles. It has 3 genomic RNA molecules. Each one is enclosed in its own particle. This virus is ubiquitous in nature. It's able to infect more than 1,200 plant species, including many native plant weed species. It's transmitted plant to plant mechanically, sometimes through seed, and by more than 80 species of aphids in a non-persistent manner. And I have this in red because I will come back to this. This is important as we talk about trying to manage these viruses. The other viruses that we are working with are 3 viruses in the genus P virus, papaya ring spot, watermelon mosaic and zucchini yellow mosaic viruses. They are flexuous rod-shaped particles. They're a single particle, not three particles like cucumber mosaic virus. They're a single RNA molecule. Their host range is much more limited than cucumber mosaic virus. Papaya ring spot virus has a slightly larger host range, but generally speaking, they're far more limited. They're transmitted mechanically from plant to plant, some through seed, but also by aphids in a non-persistent manner. So let's talk about this non-persistent manner of transmission. So as I mentioned before, an aphid will come into a field and land on a plant, and initially it will probe the epidermal cells to see if this is a suitable host for feeding. It might take a test probe and jump over to another plant or hop around a little bit, or it might just walk along that leaf and determine whether or not it's a suitable host. These viruses, when they're acquired by the aphid, this occurs in seconds when it probes an epidermal cell, these viruses actually bind to the tip of the stylet, the feeding stylet of the aphid. Even though the aphid may be taking the virus up into its body, the virus that is acquired and transmitted is on the tip of the stylet. So it can pick up that virus in seconds by probing a cell, and then without any latent period can transmit it in seconds to a neighboring plant. So this becomes a problem when we try to think about controlling this virus by managing its aphid vector. One thing about aphids too is that they reproduce parthenogenically, which means the female produces living young, and they're basically clones of them, and the populations can increase dramatically within a crop. So if you have aphids coming through and you worry about these kinds of viruses, you really have to pay attention to the aphid density and populations in your field. Now I want to move on to some control management practices that we use, and I have control here and I also have management. More likely we're trying to manage the virus because we have no way really of completely keeping it out of a field. There are three types that I want to mention. One is genetic resistance, using genetically resistant varieties, which of course is perfect for you as a grower because it requires you to do nothing but put the plant in the field. I'm going to talk a little bit about biologic formulations, and these sometimes enhance plant growth and induce resistance. And then I'll spend a little bit more time on the incorporation of production practices that delay the introduction of virus into your field. So genetic resistance, having availability of varieties that are resistant to the virus or viruses in your area is important. It's the easiest way to go. It's rare to have resistance to the viruses that may threaten your crop. But I want to mention how we look at resistance as plant virologists. So if you recall the process of infection that I mentioned, where a virus is introduced into a cell, it translates its proteins, replicates its genome, and starts moving cell to cell through that leaf, gets into the phloem bundle, moves out of that leaf and starts to invade other leaves and tissues in the plant. So in the case of a susceptible plant, one that's fully susceptible, all of those processes occur very freely. Lots of virus accumulating in all these cells, the virus moves freely, it moves freely throughout the plant, and you get high virus titers throughout the plant, and of course you have problems with impacts on yield. The other extreme would be immunity, where the virus, when it gets introduced into this cell, nothing happens. It's a non-host, the virus does nothing. That's great. What we really have is a kind of a continuum in degrees of resistance. If you were to look in the literature and study different types of resistances against viruses, there is some type of resistance that has been described that mentions every type of step in the infection process. So for instance, something that's very moderately resistant over on this end might be a virus that infects the leaf normally, kind of spreads through the plant fairly normally, but it accumulates to lower levels in these younger leaves. So that plant is basically fully susceptible, but that lower amount of accumulation of the virus in those younger leaves might put it into a type of resistance. Something that's more extreme would be a virus that can get into this first cell, It can translate its proteins, replicate freely, but then it cannot move out of that cell. It's called a subliminal infection. It's basically nothing happens. It does not move out of that cell, and it's nearly immunity. Some people would refer to this as operational immunity. And you see I have tolerance up here, kind of separated from the resistance spectrum. True tolerance means that the virus infects the plant, accumulates, and moves cell to cell as if it's susceptible. It moves throughout the plant as if it's a susceptible plant, and it accumulates freely throughout all the tissues. But that plant is able to tolerate infection, and so you have no symptoms or very, very mild symptoms, if any, and basically no effect on fruit. So using a tolerant host is very good in the short term. It's very nice because you can get a crop out and have no effect on yield. It's potentially dangerous in the long term because you have potentially the buildup of inoculum in your field. And of course, at the end of the season, it's going to go back out to your native plants or weeds that surround your field, or it's going to go to neighbor's crop or your other crop. So using tolerance, you have to be careful in knowing whether or not you have virus building up in your field. It's something to be a little bit worried about. Okay, the application of biologic formulations. The ones that we've worked with involve bacterial species, and these are soil-borne bacteria that have been isolated, and they typically are associated with the roots of plants in one way or another. Some of the treatments are single bacterial species, some are combinations of 2 or more. We've used treatments that have 30 or 40 different bacterial strains, and they're called cocktails. These bacterial species interact with the roots or live around the root system, and they live off of root exudates. In return, they can carry out one of 3 possible functions. Some of these bacteria actually induce a systemic resistance in the plant to some pathogens. It's different from the genetic resistance of, like we just talked about with genetic resistance of genetically resistant varieties. This is a kind of an innate induced resistance. Others make the minerals and nutrients in the soil more accessible to the plant, and then as a result you get enhanced plant growth. Others interfere with or somehow knock down the populations of pathogens that might be around the root itself. So what happens with these cocktails is that depending on the soil conditions, the temperature, you're hoping to have one or more of these actions taking place so that when you put your plants out there, by the way, they're applied as seed treatments or as root drenches, you'll get some induced resistance and enhanced plant growth. These are pretty nice systems. We've looked at these over the number of years. There are some that are available for organic farming, by the way, and we've had pretty good success in reducing virus infection in plants with cucumber mosaic virus and tomato spotted wilt virus. So you need to work with them to see if these systems work for you, but they are out there and they're pretty good sometimes. The last one I want to talk about is affecting production practices where we want to delay the introduction of virus into the field. And this goes back to what I said earlier, where when a plant becomes infected at a young age, it develops a more severe disease, so you're going to have a greater impact on fruit yield. That plant is a more rapid turnover for becoming inoculum for other plants in the field if the vectors come through. And as plants become infected at that young age, the impact is great, and you're going to lose more and more yield in your crop. By delaying the introduction of virus into the field, plants are allowed to mature a little bit more. Sometimes that leads to what's called mature plant resistance or age-related resistance, where the plants develop very, very mild symptoms or no symptoms at all. Sometimes they resist infection. So that's the key point in trying to come up with some schemes in production practices. One that is very effective is the use of silver reflective mulch, UV reflective mulch. Some of these mulches are remarkably reflective. I know a lot of people do not want to use plastic in their field, and that's understandable, but these are okay, approved for use in some organic systems. And the concept behind this is that as an insect flies into the field, it's going to be basically blinded by the reflective of this mulch. and it actually works. So while these plants are young and developing, if you're using this type of mulch, you will have essentially very, very few insects coming into the field, aphids or thrips or any kind of other insect vector coming into your crop, allowing these plants to grow without infection. It's very effective. Now over time, your plants will grow and start to cover up that crop, the mulch, and you'll have a reduction in the effectiveness of the plastic mulch. But by then the plants are somewhat mature. If virus is introduced, you may have a very little effect on plant growth and yield. This is one very nice approach to use. The other one is a little bit more complicated to understand. The plan here is to have inter-row living ground covers between your crop rows. So this plan has two basic hypotheses that are worked on. One is that when aphids fly into a field, in theory they're attracted or stimulated to come into the field because there's a contrast in what they see between soil and plant. The visual contrast brings them into the field and land on your plant. Now, of course, if the aphid lands on your plant and probes and it has virus when it comes into the field, you're going to get infection of your plant. The idea here is that there's a lawn of green across the field. The plants, the insects won't be so stimulated to land in the field. The other hypothesis with this is that if the aphid lands in this inter-row crop, it will test probe and in doing so lose the virus. And so that when it gets onto your test plant, it will be virus-free. We have tried this system a number of times using Lespedeza, the perennial weed, a perennial legume that we can mow. It's a nice little plant. We've tried rye, we've tried cowpea or southern pea. In one case, we actually just took a field of monocot weeds and tilled the crop into it. And we had quite good success using this approach. One thing is that the hypothesis one, where we'll have fewer aphids in the field, did not hold up. We had just as many or more in this field as in this kind of field, but we had significantly less virus being introduced to our crop plants. Now, the other good part of this is that you have soil conservation and you reduce the possible effects of erosion from water runoff. But you really need to know your virus host system here before you try this system. And I have an example here to explain why that's the case. So we tried this with tomato and pepper, and I'm going to focus on tomato here, trying to reduce tomato spotted wilt virus, which, as I indicated earlier, is transmitted by thrips. But in this case, the virus replicates in its thrips vector, so when thrips feeds, it does not lose the virus. It's not like the virus is transmitted by aphids in a non-persistent manner. We put this out, we used rye, and what ended up happening was the thrips were actually very attracted to the rye, so we ended up having significantly greater thrips densities on the plants in the rye, and as a result also greater tomato spotted wilt infection in these plants. So my suggestion here is that before you jump into this system, consider your crop and know your virus vector system that you're trying to reduce in your crop before you decide on this inter-row crop. This inter-row crop should not be a host to the virus, and you certainly don't want it to attract the vector. So let me summarize a little bit here. The plant viruses are very simple infectious agents actually, but they're obligate parasites and they remain intracellular in their host. So while they're very simple in their genetic makeup and in the way they are put together, they're quite complex because of the fact that they are so intimately associated with their host. We don't have cidal compounds to spray to stop virus infection. Genetically resistant varieties are our best approach, no question about it. If you're going to try some management efforts, for UV reflective mulch is good, the inter-row cover crop or living ground cover is good. Consider what you're using, know your virus vector system. Biologic formulations are available for organic production as well. The mission is know your system, know your virus vector system that you see is building in your area, and then integrate some approaches. Try 2 or more of these approaches to try and reduce the losses to these viruses. So that's it. Thank you very much for participating, and I will be happy to answer any questions if I can.
Okay, thank you. Yeah, we are getting some questions coming in already, and if you came on a little late, you can feel free to type in questions into your question box and hit return. I also want to remind everybody that we are recording this webinar, and it will be available within the next 2 weeks at the most on the eOrganic YouTube channel and on the extension.org website at the link on your screen to our webinar archive. If you'd like the handout of the slides, it's in the handouts section of the control panel. I know 1 or 2 people had a little trouble downloading it in Chrome, but then one person switched to Safari and had no trouble, and I was able to do it in Firefox. But please contact me if you need that and you were unable to download it. If after the webinar you have general questions about organic farming, not viruses specifically, you're welcome to use the e-extension Ask an Expert service. So now let me get to some of the questions. Okay, well, one person said it worked fine in Chrome, so okay, I don't know, but every computer is different here. Okay, one person is interested in finding out more about where you can get biological formulations and what names of some of the names of some of the products are. Do you have a list, or is there one somewhere, or can you suggest any?
Well, there are a lot of them available. You can go online and Google for biological formulations. If you look up plant growth promoting rhizobacteria, for instance, PGPR, they're usually the basis for these types of studies. But the ones that we've used are from a company called Agricin and another one called Pathway Biologics. They're two. Agricin, I think, is a big company, and Pathway Biologics is a smaller company. But if you look up PGPR and commercial products, I think you'll come across a fair number of them. And you can also, we just had a paper come out recently in the International Journal of Vegetable Science that talks about these. So they're out there. Look up plant growth promoting rhizobacteria.
Okay, great. Let's see. Did you see any positive results with roller crimper methods? I don't know if you did that at all. Maybe because there's less nitrate and therefore would be less attractive to aphids?
Well, let me just say that I have my colleague in the room here, Dr. Michael. I have not used that, but Michael is claiming he has, and he has not had success.
No, that's not one of the things we tried.
Oh, you have not tried?
No.
Have you had success?
We haven't tried it.
Oh, I'm sorry. So no, we have not tried that. We're sorry.
Okay. All right.
It was given me.
Okay. Let's see. How practical is it to induce plant resistance? Are there any materials organic growers can use to induce SAR? That's, I guess, systemic acquired resistance. Okay.
For instance, the two that we used, this goes back to the kind of the first question. The two that we used initially were actually approved for production in organic systems. So the Agricin one and the Pathway one, both of those treatments are available for use in organic production systems.
Okay. Let's see. Here's a question. Could one possibly see the young plant incur something like cucumber mosaic and then kick it, so to speak, and do well later in the year? Is that possible, or would you expect the plant to fail altogether?
Well, if it's infected very early, it's difficult for it to recover. But plants do recover from virus infections. In cucurbits, it's particularly interesting because sometimes you get the infection somewhat early, as long as it doesn't devastate the plant early on. But you could get infection early on, and you'll get decent symptoms showing up. But as the vines kick out, you'll see suddenly that the new leaves that come out are virus-free, and they have no symptoms, and sometimes they have no virus. And that's pretty nice. But what we also notice is that when they undergo some kind of a stress later on in their development, whether it's fruit production or maybe a little bit of a drought stress or something, sometimes those symptoms will kick back in again. But some plants can outgrow virus infection, depends on the plant and the virus. So you can end up with later on with a plant that's mature and has little effect from the virus.
Okay, are there any tools that you know of for identifying different viruses, like a microscope attached to a cell phone or any other type of app that might help identify different viruses?
Not that I'm aware of. Part of the problem is the virus is inside the plant, so you can't see it. It's also ultramicroscopic, so we can only see them using an electron microscope, which we cannot carry to the field. A monstrous piece of equipment. The only thing you could do would be to be focusing on the symptoms themselves. And so one problem with virus symptoms that I didn't mention is that some herbicide damage mimics virus symptoms very closely. So you have to be careful in thinking before you conclude that it's a virus symptom. You might want to check and see if you have a lot of symptomatic plants along a field edge. Sometimes it might be from a drift or treatment from an herbicide or something, but we have no way of looking at the virus in the field. That's not possible.
Okay, we have a couple questions coming in about mulches. Did reflective mulch help for TSV on tomatoes?
TSV, is that tomato spotted wilt virus? I don't know if that's...
If the questioner might want to put that in. Okay.
Okay, I don't know if that's tobacco streak virus or did they mean tomato spotted wilt virus, but what we found is that silver reflective mulch, if it's a high quality silver reflective mulch, any flying insect that might serve as a vector for whatever virus, they're deterred from coming into that crop. They do not like that reflective mulch. I mean, when we work with it, we have to have decent sunglasses on because it's quite blinding actually.
Okay.
So if it's a flying insect vector, it's going to be deterred.
Okay, did the silver reflective mulch have an effect on pollinators?
That's a good question actually. I'm not aware of much on that, but that's a good point. Of course, by the time the plants are flowering, they're somewhat more mature. So the impact of the silver reflective mulch might be less because the plant is covering the mulch. There have been studies showing that the reflective effects on plant growth, there are some effects on plant growth. So I've read a number of those, and so the UV reflective mulch sometimes can enhance the plant growth because of the reflection.
Okay, we have a couple questions on roguing and cutting out parts of disease plants. Is that a recommended practice to rogue out infected plants or parts of the plants that are infected?
So if you know it's a virus infection and you have just a few plants out there, what you're going to want to do is take a pretty large plastic bag with you and put it over the plant very carefully because you don't want to disturb the vector on that plant so they fly off to another plant. Because they may be viruliferous and they can just carry the virus to the neighboring plants. So if you want to rogue a few plants, very carefully cover it with a plastic bag, rip it out of the ground and keep it in that plastic bag. That works pretty well. I've often wondered how many you can rogue and still get a decent crop. I'm not sure what that number is, but yeah, you can do that.
Okay, which of the management practices you talked about might also have beneficial crossover effects for other cucurbit issues like mildews?
Well, let me just say this. The biologic formulations really have a much greater effect on other pathogens like fungi and sometimes nematodes. They really, if you went into the literature and studied the biologic formulations, you'll find that most of the work has been done with looking at fungal diseases and nematodes as well. So with viruses, there's not a lot available on that, but some of these things, they have a very broad spectrum approach to controlling, like inducing disease, inducing resistance.
Okay, and if you're interested in cucurbit downy mildew, on December 6th, we have a webinar coming up by Chris Smart of Cornell on managing cucurbit downy mildew in organic systems. So you're very welcome to join us for that. The first link on your screen will take you right to the registration page for that. And then also the third webinar in this series is on January 11th, and it's on management options for striped cucumber beetle in organic cucurbits. So I expect a lot of people for that one. Let's see, let's move on to, okay, did you want to say something?
No, I'm happy, thank you.
Okay, sorry. Okay, we have a couple more questions coming in. Can we bolster plant immune systems so that they can contain or eliminate viruses after infection, or is it all related to innate resistance levels?
Well, obviously if you can prime the system in advance of the virus coming in, that's going to work a little better than trying to do something after the virus is there. If the plant is young, the virus can move that plant pretty quickly. So it's difficult to think of something shutting that down once that system is going. If the plant's a little bit older, though, you might be able to treat it with something that will enhance the plant growth in such a way. By the way, some of these biologic formulations that we worked with enhance plant growth so dramatically that we found that we didn't have induced resistance, but the plants were acted like mature plants when we inoculated them. So there was like an induced mature plant resistance kind of effect. Now that was before the virus was inoculated, but I think that depending on when the plant becomes infected, the after may or may not work. And I'm not aware of what treatment that would include, by the way.
Okay, let's see. One other question about a particular product. Have you tried Immunostrips from Agdia?
Yes,
okay.
And they're pretty handy. That would be one of the types of samples I mentioned that you could take it out to the field and run a test right there. Those are very nice little handy tests. Something that growers can use, extension specialists and researchers use them a lot. They're nice.
Okay. One more question here. What size plots are you working with?
Yeah, that's a good one. We typically run smaller plots, so it's the kinds of management strategies that we're talking about there are good for smaller farms, but also when we've run these experiments, we've had lots of different treatments, so the area that we've covered has been pretty large. So I'm not sure exactly, I can't give an exact answer on that, but depending on the virus vector system you're looking at, the silver reflective mulch will work. Of course, the treatments, the biologic formulations are per plant kind of treatment, and then the inter-row cover crop approach is also something that could be across an entire large field for the most part. But the deal is, again, if you can keep the virus out while those plants are fairly young, that's really the key. And let them mature a little bit. If the virus comes in, then your losses will be much less.
Can you give an approximate scale of like what you mean by pretty large? Because I think there's kind of a question about, what might be studied in a small scale might not be so applicable for a larger scale.
I'm having to think about this.
Okay.
I'm sorry.
Okay.
The numbers aren't coming to mind right now.
Okay.
I can see the field, but I don't remember the size of the field.
Okay.
I'm sorry about that.
All right, thanks. Okay, what is the additional cost of the reflective plastic per acre, or how might somebody find that out?
Yeah, they are more expensive. Actually, those types of mulches are more expensive. I do not know offhand the difference in the cost. I'm guessing that if you have a serious threat with insect vectored viruses, it might be worth actually having that cost. But I do not have cost off the top of my head. I'm sorry.
Okay. Do you know approximately what thickness might be recommended of the reflective mulch?
Sorry, will you repeat that, please?
The thickness of the reflective mulch.
I don't know. It's a standard mulch. For a long time, they were actually just painting the mulch silver, and then companies started generating truly reflective silver mulch. But I don't know the thickness. It's a standard mulch that we've used, either white, black, or silver reflective mulches seem to be much the same for us.
Okay. Nitrogen availability in soil is said to attract aphids. Would nutrient management be a way then to manage viruses?
So if what they mean by that is that more nitrogen makes the plant healthier, I'm not sure if what they're thinking is there. So the theory is, or the theories are, that aphids tend to be attracted to yellowing plants somewhat. And it's amazing to think that virus causes yellowing symptoms has evolved in such a way that it attracts its vector. It's kind of a cool thought. But one thing to think about, I don't know if adding nitrogen will deter or attract aphids to plants, but the healthier the plant is, I'm going to say the happier the virus is to some extent, because it's going to be moving through the phloem to young developing tissues. Now that doesn't mean that you can't have a really decent amount of nitrogen and it can mask the symptoms and you can get good yield, good growth of the plant, and the virus effects will be less or lessened by that. But I think I'm not aware of anything where adding more nitrogen deters aphids. It may be true, but I'm not aware.
Okay, yeah, someone commented that the aphid's sense of direction is confused by the reflective mulch.
Yeah, I said blinded. Was trying to. They're correct.
Okay, thank you. Okay, so how long can viruses survive on something like a harvest knife?
They're on the virus. So cucumber mosaic virus is pretty unstable. So are most of the podaviruses, for instance. I don't know the amount of time that they can remain on a harvest knife. It kind of depends on, I guess, if you're again, I keep going back to these young plants. If you're moving things around to younger plants, then you're going to be risking introducing the virus. And, you also got how many virus particles there are in an individual cell. There are millions of virus particles. So if you have sap sap from an infected plant on your knife, and that sap has not fully dried or baked in the sun a little bit, there's a good chance you can transmit the virus. Now, if it's a virus like tobacco mosaic virus or one of the tobamo viruses, that's the same group there. Those viruses are stable, for remarkable amounts of time. So that's a different story. But for most viruses, I guess you could probably transmit virus for a fair amount of time, maybe half hour or so. I don't know. It's the baking in the sun, that is.
Okay, looks like that was it for the questions there. So I'd like to thank everyone who contributed questions here. And I hope you do go to the link on your screen and join us for our next cucurbit webinars in the series coming up in December and January. Thank you so much, John, for sharing your research with us, and a shout out also to Mary Kreitinger, who helped organize this series. And thanks to everyone for joining us.